Hi everyone. In last lecture series, we have seen in detail about the ion exchange chromatography, and in today's lecture, we can see in detail about the size exclusion chromatography and its principle and working mechanism. Coming to the introduction of size exclusion chromatography, it is also known as gel filtration or gel permeation chromatography, whereas it involves a porous structure of material that have a minimum size of pores to elute different sized particles, whereas this size exclusion chromatography used to separate biological molecules and to determine the molecular weights of different compounds, and also it can give the molecular weight distributions of different sized polymers, whereas this can be used to separate different macromolecules and also industrial polymers of different sizes. Whereas if you are going to use solution as a mobile phase that can pass through the column, then the column is called gel filtration chromatography. If you are going to use organic solvents as a mobile phase through the column, then it can be called as gel permeation chromatography. The separation in this kind of chromatography is called as fragmentation of different kinds of proteins. Sizes of different pores in the beads, that is acts as a supporting medium inside the column, can be determined with the help of exclusion limit. Coming to the principle of size exclusion chromatography, a mixture of compounds that is mixed with a liquid. This liquid is called mobile phase. When introduced into the column, it is react with the supporting medium which acts as a stationary phase. Here the mobile beads are used as a stationary phase, whereas this beads having different size of pores to elute out different sized compounds. Here in this size exclusion chromatography, the beads that contains different pore sizes acts as a traps or sieves, whereas this traps very small kinds of particles, that is very small sized particles inside the pores of each bead, whereas this allows large sized particles out of the trapped beads and gets eluted first. Likewise, the size exclusion chromatography first elute out large sized particles and it traps the small sized particles and eluted later, whereas this size exclusion chromatography helps to replace the compounds with different time periods of different size of compounds. Coming to the working mechanism of size exclusion chromatography, here it involves 4 different steps, whereas in first step we have to make the column fit, that is we have to pack the column with different pore sizes of beads. These beads are made up of semi-permeable membrane so that it can trap so much of particles, macromolecules and different sized proteins inside the pores of beads, and it can allow the sum of the biomolecules outside the pores, so that we can separate out small size and large size particles. And in next step, we can pour the mixture of protein molecules at the mouth of the column, whereas these molecules will enter slowly into the column and trapped inside the beads that present as the supporting medium, whereas large size particles that may or may not be entered into the beads can elute out first at the higher rate. Whereas in last step, when we are adding some of the mobile phase as an elution buffer, here based on the elution buffer capacity, the beads that contains or trap some of the small size particles can also eluted out at the last. During this elution, we can separate out different size proteins, whereas very small size proteins gets trapped inside the beads so that this remains inside the beads for long time, whereas medium sized proteins gets move into the other beads and travel through the column, whereas large size particles cannot enter into the beads or pores, whereas this large size particles enter the void space present between two different beads and gets eluted out first at the large rate. Then the medium sized particles eluted out at the second stage, and also when we are going to elute out very small size particles, we have to use elution buffer for separating out or for purifying very small size proteins. Let us see in detail about different components of size exclusion chromatography, whereas this includes stationary phase, mobile phase and different types of pumps and also detectors and different types of columns. Coming to stationary phase, the stationary phase is nothing but it is semi-permeable membrane of porous beads, whereas these porous beads can be of different size of ranges, and here the beads are mainly made up of different polymers and it can be made with different cross-linking stages, whereas the degree of cross-linking should be controlled for the different range of sizes of different pores. In this stationary phase, we can set very small size pores for the proteins that to be desalting out and for different protein purification, whereas the intermediate sized of pore sizes can be set for different biomolecules to be separated out, and very large size molecules of pore sizes of beads can be set for separating out large sized or macromolecules of different mixture sample. Two different types of stationary phase are used commercially, that includes dextran, that is Cephedex, and also dextran polyacrylamide and dextran polyacrylamide, which is cephacryl. And these kinds of stationary phases can be available at different size ranges for different types of protein separation. Coming to mobile phase, the mobile phase forms an important component of SEC. Here, the mobile phase forms a liquid part that dissolves many biomolecules or macromolecules of components that to be separated, and this can be mixed with buffer to form a perfect mobile phase, whereas the components that gets mixed with this buffer is called sample. And here the mobile phase can elute out different polymers at room temperatures. For example, tetrahydrofuran, dimethyl and chloroform forms as an example for the elutant. Here, when we are choosing the stationary phase and mobile phase, this should be in accordance with each other. For example, if you are choosing the supporting medium as stationary phase, for example as synthetic elastomers, we have to choose toluene as a mobile phase as a solvent. And if you have to choose PVC or styrene as a material as supporting medium, we have to choose tetrahydrofuran as a solvent. Coming to the next component, that is columns used in the chromatography. Here there are so much of commercially available columns have been designed. So based on the lengths of different columns, the column may be analytical usage and for preparative usage, whereas many columns have been designed based on different length sizes like 25 cm, 30 and 50 based on the laboratory purposes. And nowadays they are using a narrow bore sized columns with 2 to 3 mm size, which can be used for very fast and accurate type of chromatography. Coming to the next component, that is pumps used in the chromatography. Usually pumps are used to pump out different compounds that is injected into the columns. Here these pumps should have a highly constant flow rate throughout the entire chromatogram, because these pumps are highly depends on the flow rate concentration. And here the pumps that have a 0.1% of error can cause up to 10% error in the molar mass of the samples to be separated out. So we have to concentrate on the pump's flow rate accurately. And in this, here we are going to use reciprocating pumps and syringe pumps for various separations based on the size. Most of the pumps in this can reproduce flow rate up to 0.2 to 0.3%, whereas here inline filters are used in solvent reservoirs to avoid damages that happens through the solvent flow into the pump head and other valves and also in pump seal. Coming to a specific type of component, that is detectors. This detectors forms an important part in SEC because this can separate out different proteins at different concentration time period. Here the detectors used are concentration sensitive detectors and molar mass sensitive detectors. In concentration sensitive detectors, we have bulk property detectors that can be detected using refractive index detectors, and solute property detectors that can be used with UV adsorption detectors. And also we have evaporative detectors that can be detected through evaporative light scattering detectors. In molar mass sensitive detectors, we have light scattering with low angle light scattering detectors and multi-angle light scattering detectors. And also we can do detection using viscosity detectors. And some other detectors are FTIR, that is Fourier transform infrared rays detectors, and also mass spectroscopy can also be used as a detectors. Coming to the advantages of size exclusion chromatography, here we can use very small amount of sample since the column size is very small, and there is no mobile phase loss, and due to this we can set a flow rate at constant rate. By using flow rate at constant rate, the separation can be done at short analysis time, and there is well defined separation with narrow band and good sensitivity. Coming to the application of size exclusion chromatography, here there are vast applications available. That includes the chromatography can be used for protein separation, protein purification, and also for copolymerization studies, and this can also be used for desalting studies. Whereas this can also use to study or determine different molecular weights of different proteins. In this, we can also separate out different proteins, biomolecules and rubbers with different sizes, and also this can use to determine the quaternary structure of different proteins, whereas this can be used to separate out different polymers based on their sizes, and in this we can also separate viruses and RNA of different biomolecules. Let us come to the end of this session. I hope you have all understand in detail about the size exclusion chromatography and its principle, working mechanism and different components, and also advantages and application of size exclusion chromatography. In forthcoming lecture series, we can see in detail about the principle and working mechanism of hydrophobic interaction chromatography. Thank you.